Morning, everyone. Welcome, everybody. We are so glad to have you with us today for a virtual meeting with one of our scientists. Thank you so much for joining us. My name is Rachel. I work with the Education and Natural History Museum. Today, we're going to be meeting with a special guest from our Research and Collections Department. The Natural History Museum is home to 35 million objects and specimens. Our scientists use these collections to conduct research about people and nature, past and present. Each scientist has a special area of focus for their research, but together, they're helping to build the history of life on our planet. In just a moment, we're going to hear from Dr. Bill Ludt, Assistant Curator of Ichthyology, and ichthyology is the study of fish. Dr. Bill Lut's research aims to understand why there are so many fishes on our planet. Central to his research are museums, specimens, and the data associated with them. We'll talk about the importance of our ichthyology collection, as well as some of the slimy adaptations that some of these specimens in our collection have. So I'm going to go ahead and stop sharing my screen so we can meet our presenter today, and you'll see me again in a little bit. Hi, Bill.
Hi, Rachel. Thanks for that introduction. I'm happy to be here today to talk to all of you, and hopefully some of you are excited as much as I am about some of the fishes on our planet. And you are going to hear me say the word fishes as a plural for fish instead of just fish, which you might have been taught in school. But as scientists, we use fishes instead of fish to refer to multiple different species of fish. So don't get confused if you hear me say that word moving forward. Today, let me start sharing my screen because I have a lot to talk to you guys about with what we do at the museum and what I specifically do with my research. So, fishes, slimy fishes today too. Before I get into the slimiest fishes, I am going to tell you a little bit about our collection here, about how I use use the collection for my own research. But before I get into any of that, what I would like to start with today is actually talking a little bit about kind of how I got to be where I am today. So how does that path work to become a scientist, and especially a scientist that works at a museum? So I grew up actually not too far from LA, about 8 hours away or so. I grew up in southern Arizona, where the landscape looks like this, saguaro cactuses and lots of spiny, prickly plants and not much water. Not an area where you'd see, a lot of fish. And some scientists get to their careers in science because they know from a very young age exactly what they want to study. And maybe some of you are like that too, where you really are interested in one certain type of thing, and that's great. But not everyone is like that. So if you don't have this passion for one specific topic quite yet, or don't really know what you want to be when you grow up, that's perfectly fine. I certainly didn't know until college. Growing up in southern Arizona, I actually didn't know much about fishes at all. And so my first experience even for many fishes were on a couple of trips that I made growing up. I got to go to San Diego one year where I did some snorkeling on the beach, and that was really fun. And I really liked looking at the fishes. This is when I was much younger. But it really wasn't until college when I actually decided to get scuba certified that I really started getting into wanting to know more about fishes. And those trips were actually down in Mexico. This is a picture from some of my earliest diving trips with some old friends. And it was these trips that really got me thinking more about what I was seeing underwater, primarily because I didn't know what I was looking at. I would go scuba diving, and it was a lot of fun, and it was a completely different world underwater. But when I would look around me, I actually didn't know what any different types of fishes I was looking at. And so for some reason that clicked with me, and I just became insatiable with wanting to learn more and more about the fish on our planet. So that's what led me to pursue an education where I learned more about fishes and more about museums. And museums really hold a special part or they really are a central role in our understanding of life on our planet. So today, as Rachel introduced me as, I work here at the Natural History Museum of Los Angeles County. I'm the assistant curator of ichthyology. If you haven't had an opportunity to come to our museum before, this is what it looks like on the outside. Or if you have been to our museum, it might have been a while since we are currently still closed to the public for our indoor spaces. On the outside, we have these wonderful nature gardens, and we show banners of our current exhibits that we have on display. And on the inside, when we are open to the public, you can see that we have various dioramas that show life on our planet in different areas, or show life that's not on our planet anymore, but that used to be, such as our fossil collections and our dinosaurs. And this is kind of what a lot of people think museums are for, just showing these displays. But actually, a lot of museums, like our own here in LA, are also research museums. And there's a significant portion of the work that goes on behind the scenes. And this is where I work. I don't work out in the public area. I work behind the walls in our collection spaces. And at the museum, we have a lot of different types of collections. Maybe you've seen some of the other specialists or curators talk about their own research in a couple of these Meet the Scientists in the past, but what we have are large collections of individual specimens that have been collected at different places and at different times. So I'm showing, parts of our entomology collection, parts of our bird collection here, and in the upper right, you can see fossils in our paleontology collections. And I like to think of museums actually kind of like a library. And in a library, you can find different books that were from authors that published them all over the world. And they also come from different times. So you can see some books that were published several hundred years ago, up to books that were published just a couple days ago. Some of these authors are no longer with us, and we can only learn about them or the way that they thought through their books. And a lot of these books look different sizes, different colors. Museums operate in a very similar way. Instead of books, though, we have specimens. And we have specimens from all over the world. We have specimens that were collected at different times. So in our own fish collection here, we have several specimens that are a couple hundred years old. We have, unfortunately, animals in museums that are no longer with us. So extinct animals, and the only way that we can study them now is by using museum specimens that were collected when they were around, or in the case of fossils, like understanding dinosaurs, in specimens that we dig up from traces that they left behind. And so museums very much act in a way that libraries do. For research, you would go to a library to learn more about a topic. Scientists come to museums to learn more about animals. There is a difference between libraries and museums. A lot of libraries, you can scan pages and make digital copies of things that you can access from your own home computer. That's a little bit harder to do, although I will talk a little bit later about how we are striving to make some of our specimens more accessible. So I am the curator of ichthyology. I study fish, or fishes, as you'll hear me say. Here off the coast of California, we have some very notable fishes. They come in a lot of different colors and sizes. So on your upper left, you have the sheep's head. On the right, you have the Garibaldi, which is our state fish, a very bright orange fish that you can find if you snorkel or dive around here. Bottom left, you can see a sarcastic fringe head, which looks very scary, but they are pretty small fishes. And I love fishes just because they are so extremely diverse in what they look like, where they are, and what they do. So these are just some examples of our local fishes, but if we look more globally, look at fishes all around our world, we can see fishes are found in freshwater, they're found in saltwater, they are found all the way at the very bottom of the ocean, all the way to alpine lakes several thousand feet above the ocean too. There are big fishes, like in the center, the whale shark, that's the world's largest fish. And there are very small, tiny fishes as well. Some are colorful, some are not, and they just come in all sorts of shapes and sizes. Altogether, there's over 35,000 accepted valid species right now of fishes. And that's more than birds, more than reptiles and amphibians, and more than mammals all combined. So if we talk about vertebrate diversity, vertebrates being animals with backbones, kind of like us, fishes are the most diverse out of all of vertebrates. So we don't actually, one common misconception is that our fish collection is full of fish tanks of live fishes. And it's not. This is an image of our fish collection. As you can see, it's just a bunch of rows with shelves. We have a compactor system, we call it. You'll see the handles that we can use to turn the shelves so we can save some space. And so we can keep our shelves closed just in case we have earthquakes to protect the fishes. On these shelves, what we have are a lot of jars. And again, these are not live fishes. Like a library has old books that were collected or that were published many years ago. We have a lot of fishes that were collected many years ago, as well as fishes that were collected recently. And the majority of our collection are adult specimens that are stored in alcohol. And so again, we have some really strange fishes in our collection that might not look like you might expect a fish to look like. So upper left is a great example. It's a shrimp fish. And the lower left is a bellowfish. So very weird bodied fishes from all over the planet and collected at many different times. In addition to these adult specimens, though, we keep a large variety of different types of specimens in the collection too. And all of these different types of specimens help researchers study various aspects of what a fish does or how it lives. So we have a large skeletal collection, allowing researchers to look at individual bones of a fish. This also helps people that do anthropology, that dig through remains left by ancient civilizations. They can compare bones that they find in those digs with bones that we have in the collection from modern fishes and figure out what people were eating at different time points. So we keep a large skeletal collection. Sometimes people want to look at the skeleton inside the fish and not actually look at individual bones. So we have a large collection of what we call cleared and stained fishes. These are fishes that have had a lot of their tissues digested away with enzymes, just like the enzymes found in your stomach. And then we stain the bones different colors, and we also stain cartilage. And this allows researchers to look at how bones interact with one another inside the body of the fish. We also have a lot of larval fishes in our collection. This is a baby filefish, and many larval fishes don't look actually at all like the adults. Larval fishes or baby fishes serve a very different purpose in the fish life cycle. This is actually how many fishes disperse. So a lot of fishes don't really swim super long distances as adults. Of course, there are big exceptions to this, such as tunas and swordfishes and things like that, that do swim very long distances. A lot of fishes will actually only disperse as larvae, as baby fishes. So we have a large collection of those, so we can understand which larvae go to which adults. And we have a large otolith collection, which are sometimes called ear stones or ear bones. These are found in the head of the fish and can actually be used to understand where fishes are at different life stages, and we can use these to understand how old fishes are, because just like tree rings that add on a new ring every year as the fishes age, otoliths do something very similar. So you can actually figure out how old a fish is based on their otolith. Now, in addition to all of this, we do have digital content associated with some of our specimens. And I did say that museum collections are a little bit different than libraries in the sense that it's hard to scan fishes or it's hard to scan museum specimens and let them be available to the public, or at least a little bit harder than scanning a book. But we are pushing forward, and we have new technologies such as CT scans that allow us to look at the inside of fishes without actually having to dissect it or without having to damage the specimen. So we can put these online so that researchers all around the world can look at our specimens from their own home or from their own computers. And we are pushing forward with other digital ways of making all of our specimens available. So that's kind of just what our collection is like. And I use this for my own research. And my own research is a little bit different than the specimens that I've been showing, in that I focus on 2 or 3 main areas. Although as a museum scientist, we end up doing a lot of various projects all over the world. So some of my projects might just be doing surveys of fishes in a certain area to see what fishes live there. But my own research really focuses on a couple main things. One of those is understanding how fishes are related to one another. And that is a field called phylogenetics. And one project that I've worked on looks at surgeon fishes. These are a couple examples of those. You might be familiar with them because of certain popular movies. In fact, on the upper left, that is a real life example of Dory from Finding Nemo. So one of my projects, or some of the work that I've done, has looked at how different surgeon fishes are related to one another and where they live. And so this is a diagram that shows how a particular type of surgeon fish called saw-tail surgeon fishes, how they're related to one another. And you can see on the right, those 3 species, because they cluster closer together, and those lines that connect them are more direct, you can see that those are more closely related to one another. And we can use this information, or I use this information, to understand where fishes are and how they got there. And in particular, my research focuses on fishes that are called anti-tropical fishes. These are fishes that live just outside the tropics. And I'm showing the tropics in red on this map. And this is tropics just by temperature, not by latitude. And you can see, so I study fishes that are right above the red area and right below the red area. And I study these fishes because they are influenced a lot by climate and by environmental factors. And today on our Earth, obviously, we do know that there's a lot of rapid climate change. It's in the news a lot. And I actually look at how past climate change influence where these fishes are and how they got to be where they are today. So for this, I use museum specimens again, but I use information associated with where those museum specimens were collected. So every museum specimen has information on where it was collected, and from that I can get temperature, salinity, and other environmental variables. And we can use this to create models to figure out where these fishes should be able to live and how that's changed over time. And so that's just a brief overview of a couple of the things that I study. So phylogenetics, how fishes are related to one another, and biogeography, which is the study of where species live and where they have lived in the past. But today we're also doing slimy fishes, because this is in partnership with Nickelodeon, and we are doing the science of slime. And so let's talk about some of slimy fishes. Now, you might have gone fishing in the past, and you might have caught fishes that look like this. These are, bluegills and other sunfishes, largemouth bass or crappies. These are very popular freshwater sport fishes, or maybe you've gone saltwater fishing. If you've ever gone fishing in general, though, and you've held a fish, you can tell that it's pretty slimy. And this slime serves a couple of main purposes. It really helps fishes move through the water, so it reduces drag. It has antibacterial properties to help keep the fishes healthy, but some fishes use slime in very different ways and very remarkable ways too. So a fish that you might be familiar with, because you might have it at your house, is a betta. Bettas use mucus or slime to create these bubble nests where they put their fish eggs to develop. So these fishes can actually use slime in a way that's outside of their body. So they just produce these nests. And dwarf gouramis do this as well. So another popular aquarium fish that you might be familiar with. Another popular fish that you might have at home is a discus. It's a type of cichlid. On the right, you can see a head-on view of a discus with all these baby fishes all around it. And this is because discus fishes use slime on their body to feed their young. So their young will actually eat the slime off their bodies to help sustain them right at the beginning of their lives. There are other fishes that use slime that you might not be so familiar with. One example is this African lungfish. There are actually 4 species of African lungfishes, and some of these fishes live in areas that actually dry up. So the streams or ponds that they're in get completely dry during parts of the year. And what these fishes do is they'll burrow into the mud and they will create a mucus or a slimy cocoon that they will actually hibernate or estivate in for up to a year between wet season and the next wet season. And so these fishes use slime in a very different way than some fishes that you might be familiar with in your home aquarium. And there's also marine fishes, of course, that use slime. So one great example is a parrotfish. Some parrotfishes, when they sleep, they create a mucus cocoon that you can kind of see here. It's hard to see because it's transparent, but you can see some of the sand sticking to it. So parrotfishes will go into cracks and crevices or under corals, and they will secrete this cocoon that will protect them from any bugs that want to, or any marine invertebrates that want to feed off of them while they're sleeping. But the slimiest fish of all, of course, is the hagfish. These are deeper water fishes generally. There are around 80 or so species of hagfishes all around the world. They look like an eel, but they're not related to eels at all. In fact, these are some of the earliest diverging fishes. So they've been around on our planet a very long time. They don't have jaws, actually. They're called jawless fishes. And one thing that they do, though, to prevent animals from attacking them is they produce slime, and a lot of slime, copious amounts of slime. So one fish can produce a lot and lots of slime. And the purpose of this is to deter predators from attacking them. So a fish will try to eat them. They'll produce all the slime, and the slime will actually gum up the mouth of the predator, maybe get into the gills if it's another fish trying to eat it, and the fish trying to eat it will just have to let go of it because it's just not palatable. It's not edible at that point. They produce so much slime that, in fact, a couple of years ago, there was a car that had hagfishes for research purposes in its trunk that got in a car accident. And you can see just the amount of slime that a couple hagfishes can produce. It is absolutely astounding. So these are definitely the slimiest fishes on our planet. Although, like I said, all fishes have some sort of slime, but there are some really cool examples of slime in fishes out there. I think I've gone over my time with talking, but if you want to know more about fishes, you can either see our website. I also have an Instagram account, if any of you are on Instagram, called Planet of Fishes, where I post pictures of fishes from our collection or fishes that I come across when I'm out doing work in the field. But I'm happy to move on from this and answer any questions that you guys have for me.
Thanks so much, Bill. That was so fascinating. I loved seeing all of those cool collection images and learning about lots of different types of fishes and their slime. We have a lot of great questions, so I'm going to jump right into it. You started by talking a little bit about your kind of pathway through your career. Leslie was curious, how long do you have to study for to become what you are right now, an assistant curator at the museum?
So to become a curator at a museum, you typically need a PhD. So that means you have to do an undergraduate degree, which can, take around 4 years or so is about average. And then you can either go straight from undergrad to PhD, or you can do a master's first and then a PhD. So you're looking at a lot of time in school to become a curator, but there's also other careers and other fields in science that don't take as much schooling. So if you don't want to be in school as long, there are so many different jobs at a museum like ours or at other institutions where you can participate and work in science as well without having to be in school for so long too.
Awesome. And we had a question from Carmen. I thought this one was kind of fun. Are you ever grossed out by what you see sometimes in the collections or in your research?
In the collections, I'm not as much grossed out because a lot of those fishes are preserved and kind of clean at that point. There are some fishes that we collect out in the wild, though, that are really gross, either because they're really slimy or they smell bad, or they're just like, yeah, fishes, many of them I love to death and they're fascinating, but some of them can be pretty gross when you're actually working with them.
I can imagine. Speaking of kind of slimy fishes, Shakina was kind of curious, why do fish need scales or slimy skin? And you kind of spoke a little bit to this, but can you clarify kind of the purpose of that slime again?
So scales help protect fishes. In fact, some scales, there's different types of scales. And one type of scale is called a ganoid scale that fishes like gar have. That is basically like armor. I mean, it is hard to puncture. And so scales help protect fishes. And slime, though, can do a bunch of different tasks. And I think some of the most important things that slime does is it has that antibacterial properties. So it can really help a fish, avoid infection if they get a small gash. But it also will help if there's fishes that get out of the water, it helps them not desiccate. So it helps them not dry out. It can help fishes in the water if like flounder and other flatfishes that are right on the bottom, they're typically pretty slimy, and it helps them actually go over sand and other abrasive materials without getting hurt. So slime can work in many different ways for fishes.
Very cool. We were talking a little bit about the collections, and we had a couple questions for that. Aiden was curious, what is the difference between what you can learn from a live specimen and a dead specimen versus a dead specimen?
So there's definitely pros and cons to having specimens on the shelves versus having a live fish in the hand. You can't tell things like behavior from a museum specimen typically. So that you would have to be out and actually observing the fish. Behavior or things about physiology. So if you wanted to understand the metabolism of a fish or something like that, that has to be with a live individual. But the counter to that is museum specimens let you look at the inside of fishes. So you can look at how organs are arranged. You can look at the skeleton. You can do all that sort of thing that it would not be ethical to try to do that with a live fish. So there's pros and cons to both, and to have a really good balanced view, you need to study both.
Very interesting. Good to keep in mind. I'll do two more questions for you. George was curious, do you know of any extinct animals that we have in the collections at the museum?
Yeah, we have a couple extinct fishes in the collection. So at the museum in general, we have a lot of extinct animals. We have very extensive paleontology collections, so dinosaurs and things like that. But in terms of non-dinosaurs and maybe recent extinctions, we have a couple fishes in the collection that fit that category. One is the Chinese paddlefish. That was actually just declared extinct this last year. We do have some specimens in the collection that were collected many decades ago, so a long time ago before they were really endangered. And we also, for a more recent or a more local type fish, we have a couple thick-tailed chubs. These are freshwater fishes that were found that are found in Colorado and Arizona. And those are presumed extinct as well. And we do have a couple of those in the collection from, again, a long time ago. Those date back to the 1920s and 1930s that we have specimens of.
Wow, that's crazy. Okay, and I have maybe what will be the hardest question of this whole session for your last one. Ariana was curious, what is your favorite fish?
Oh, it's always so hard. And this question always comes up, but there's so many different fishes. That my answer keeps on changing on this. It really depends on what I've been looking at in the collection kind of that week. But there are some really, I'll name a couple that I really like. One that I really like is called a lump sucker. It's a small fish that just looks really cute. You can look it up on Google. And you'll see examples of why it looks so cool and why it might be one of my favorites. Other fishes are spook fishes or barrel eye fishes. I think are really cool. These are fishes that some of them can actually see through the top of their skull. So their skull will actually be transparent. They are deep water fishes, and they can look in front of them, but they can also rotate their eyes to look up above them. And they can actually look through the roof of their skull, which is just mind blowing that they can do that. So those are a couple of my favorites. We do have good examples of those in the collection here, so I can actually look at them up close. But there's just so many fishes. It changes all the time.
I'm sure. Thank you so much, Bill. So appreciate your time and answering all of these questions. Thank you for your time today.
Yeah, it was a lot of fun.
Awesome. All right, I'm going to go ahead and close us out of our program this morning. Thank you to all of our students and teachers for joining us today. We had so much fun learning about ichthyology, the study of fishes, and all the different fish that Bill studies at the museum. We also got to learn about some of our slimier fishes in partnership with Nickelodeon. Super fun. We'll have all of these videos, including the recording today, on our YouTube channel for you to watch later if you'd like. So you can catch this recording and others at youtube.com/NHMLA. Thank you so much for joining us. We hope to see you again soon. Have a great day.